All right, welcome to class. So people in the Thursday section, let me update you, and we'll take a vote on this at around 4. Okay. So the proposal, which was overwhelmingly voted for by the Tuesday section, is to split next week's exam. Okay. So next week's exam is endocrine and muscle. I personally feel it's way too much on one exam. Way too much material on one exam. I don't think that it's at all good. And I've already written the exam, so I'm gonna have to go back and redo the exam if I need to. But the proposal is to have just the muscle exam, 50 points next week, and have the endocrine exam the week after. Okay, we don't have any exams the week after. So split it into muscle and endocrine. Endocrine is way too important, okay, to learn in a hurry. Am I making sense?
Yes.
So I will ask for you to think through this and give me a vote at 4 o'clock. Okay, we'll talk. As soon as I go over the material, you'll get an idea that you do not want to take this next week. Okay? Okay, so that should give you an idea that you really shouldn't be taking this next week. And I want people to do well on the exams. Okay, I mean that's the whole point. So I don't mind going back and restructuring the exam. Probably it'll take me half hour to restructure it. It's not that bad. So, and I'll be happy to do it. Okay, so again, 4 o'clock, we'll take a vote on whether we should split. And again, I want a unanimous vote. Even if one person opposes this, then it's a problem for me. Okay, I cannot do this. I cannot split the exam. So I need everybody to okay this before I decide and say, okay, I'm splitting this exam. Okay, so the proposal is next week you'll have an exam as scheduled, but it will only be a 50-point exam on muscles only, just muscles. And the week after you will have an exam on endocrine, just endocrine. Okay?
Yes.
Okay, and then you have spring break, and then we come back, and then you have an exam on cardiovascular. Okay, but you have a week to study for it. Okay, not an issue. And in fact, I will open the cardiovascular module. It's ready to be opened. I'll just open it. And so that way you have access to all the videos if you want to learn it ahead of time. Everybody got that?
Yes.
So 4 o'clock, we'll take a vote on this. And I know you don't like notes on the board, and you know that I will try and go as slow as possible. But the problem is I can't remember all this. Okay? If I'm going to sit and write, you're going to have 25% of the material missing. And I will only do this for endocrine, okay? All the others will do it as before, where I write the notes. That I don't have a problem with. But endocrine, I'm not going to remember all this. This is a lot of information. So if I don't mention it, the problem is it'll be covered in the video, it'll be on the exam. Make sense?
Yes.
And endocrine is way too important for me to forget anything. Okay? Because you're going to use this and apply this in every chapter that's coming. So endocrine and nervous, that's why we did them so ahead of time, right? Because you're going to apply this everywhere from now on. Are we clear?
Yes.
Yes.
So, yeah, so I apologize ahead of time for doing this. It's not that I don't want to respect your wishes, but this particular chapter is too information dense for me to remember and put it all up. Does that make sense?
Yes, yes.
So even though this is my field of study, and I probably could reproduce 95% of the material blindly writing, okay? I could. I wrote this, right? But I wrote this under very calm environment, okay? Not while I'm talking. I'll tend to forget, okay? So I don't want to do that. So I want to go through this. If you need me to slow down, if you need me to repeat anything, just say so, and I'll be happy to repeat and explain this again, okay? Mm-hmm. So the endocrine system, the word, again, you must understand the word, right? The word crine, okay, and we'll do half of this lecture during lab. So we're going to be doing a 2.5 hour lecture during lab on Thursday. So because lab itself is only 5 minutes of looking at slides, okay? So the rest of the time we'll be spending in lecture, and you'll have 2 weeks to study this material, which I think is a good, decent amount of time to master this material, okay?
Yes.
All right, so crine, the word crine means secretion, okay? And there are 2 major types of secretions. You have exocrine, And exocrine means getting things out of the body, and those include digestive enzymes. Now, you can be surprised at how come digestive enzymes are secreted out of the body. Aren't they secreted into the intestine? The inside of the intestine is technically outside of the body. I want you to know this. From mouth to anus, it's a hole. H-O-L-E, right?
Yes.
From mouth to the anus, it's a continuous hole, okay? And the analogy here is if you take a donut that has a hole in the middle, right?
Yes.
The inside of the donut is still air, correct?
Yes.
It's outside, so it's like that. So imagine a stack of donuts, right? From the mouth to the anus, right? That's what the intestinal tube is. So the innermost part of our body is bacteria and air and outside stuff. Okay, so we secrete enzymes into those tubes, so that's technically secreting outside of the body. So it qualifies to be exocrine. Are we clear? Digestive enzymes are qualified to be exocrine. And endocrine is secretion directly into the bloodstream. Endocrine is secretion directly into the bloodstream. And the most important characteristic of this is that there are no dedicated ducts, and the word duct means tube. There are no dedicated ducts. Now, think about sweat, right?
Yes.
Sweat. Do you have a sweat duct?
Yes.
Yes. So that's exocrine. Okay, if you think about semen, semen has a big duct system, right?
Yes.
So semen is exocrine. Also, technically, it's called xenocrine because you're taking it from one body and depositing it into a foreign body, right?
Yes.
So that's actually called xenocrine, but still qualifies as exocrine, right? Because it goes through tubes. Hormones don't use tubes. Hormones are too important. We don't want to limit the secretion of hormones to tubes, so we don't use tubes. So these are ductless glands. So endocrine glands are ductless glands. This chapter is very, very important for your TEAS exam, okay? Big deal for your TEAS exam, okay? They're covering more and more and more endocrine nowadays in an attempt to weed off students. Okay, so endocrine is a big deal. Lot of teachers don't do a lot of endocrine in their class, but I will. Okay, this is important. Hormones are defined. There's a 5-part definition. Before I go there, are exocrine and endocrine glands clear?
Yes.
I'll give you a minute to copy. These notes are up for you, by the way. Yeah.
Why the endocrine glands are ductless?
Uh-huh.
Ductless, why?
There are no tubes.
No tube?
The hormone is directly secreted into blood. There's no dedicated tube. So for sweat glands, you have a dedicated tube going from the gland to the outside, right?
Yes.
Since these go into blood, right, you don't need tubes.
Oh, okay.
There's no dedicated ducts. I don't have the who? It should say 112 endocrine notes.
We couldn't find it.
Maybe I didn't put it for the Thursday class. Thanks for letting me know.
I see it on the Tuesdays.
Not on the Thursday.
That's my favorite class.
Sorry, Doc.
I will cry.
Let's see. I did say copy. I don't know why it didn't go through. And look, I wrote your exam, right? Where is that? That's not here either. That didn't copy either. Jeez. Don't understand this at all. Okay. Modules. Well, see, the problem is it copies, but it doesn't publish. Do you see the hormone table up there?
Mm-hmm.
I have to do this every time. This is ridiculous. Okay. Can't move this over. Okay, well, let it stay there for now. Let me go there. Well, let me see if at least my, yeah, see your exam's here. Lecture exam too, I wrote it, so
No.
This is.
I'll split it.
Should say Let's be- let's
No.
see.
Yeah.
Check refresh.
So you will need to refresh your canvas. It'll take a minute or two to copy, but it's there, okay?
Maybe give it a minute.
Yeah, just give it a minute, okay? It should be there. So what is the definition of a hormone? Surprisingly, we don't have a good definition for a hormone. So one of my dearest friends and my mentor, he came up with a 5-part definition for a hormone. Again, you won't find this in any textbook, okay? And I find this to be a very comprehensive definition. So it's a 5-6-part definition. A hormone is a chemical. Everybody agrees with that?
Yes.
Okay, I need you to participate, or I'm going to go rocket speed. Your participation slows me down, okay? So participate, say something. So, especially Thursday class, you didn't have a 3-hour torture chamber with me. So, yeah, I need you to talk. Okay, so hormones are chemicals that are synthesized by source cells. Cannot give an easier name to that, right? Source of the hormone, right? Source cells. Everybody understands that?
Yes.
Okay. Then some hormones, especially protein hormones, are usually stored in the source cells. Again, please note that only protein hormones can be stored because steroid hormones are hydrophobic. They get out by simple diffusion. You cannot hold them. It is impossible to store steroid hormones at the source. This will be on your exam. Okay, please note, steroid hormones cannot be stored at source because they are hydrophobic. They leave by simple diffusion. So as soon as you make them, they get out into the bloodstream. Are we clear?
Yes.
Okay, and protein hormones are always stored. The main reason for that is protein hormones are essential for life. Okay?
Yeah.
So for example, you have hormones that control blood pressure. If your blood pressure falls below dangerous levels, do you have a lot of time to be screwing around and make new hormone?
No.
No. They need to be released immediately, correct?
Yes.
So what you do is you have a reserve of these hormones, and as soon as you get the 911 signal, you release these hormones. Are we clear?
Yes.
Steroids don't do that. There is no steroid that, the slowest steroid is 3 minutes, okay? Minimum, okay? Steroids take a long time to work. Are we clear?
Yes.
So they don't need to be stored. You don't need to keep reserves and things like that. There's no evolutionary need, okay? But proteins, you do, okay? Because there may be life-critical parameters that are controlled by protein hormones. So they're secreted into blood. That's why it's endocrine. Endo means inside of the body. Blood is as inside as it gets. And they travel long distances. For example, a hormone secreted by the liver travels to the brain, correct?
Yes.
So long distance travel, okay, they travel all over the body, and that is termed as systemic. You already know this term. Systemic versus local. Local means in one particular spot, right? This is systemic, meaning travel all over the body. They bind a specific receptor protein. By this point, you already know this, okay? You should know this. We did this with indirect neurotransmitters, right? They bind a specific receptor protein. That's being redundant, right? When I say receptor, they're automatically specific.
Yes.
All proteins are specific, right? So these proteins, receptor proteins, are present in what's called target cells. I want to really explain this because this is important. A source cell secretes a hormone, correct?
Yes.
And then the hormone travels, right? All over the place. And then it finds a target cell. Now that automatically, logically implies that not all cells are targets. Correct? If I'm saying a target cell, that means it's very specific, right? So not all hormones target all the cells in the body. That doesn't make any sense. Correct?
Yes.
For example, estrogen, when estrogen is a hormone, when estrogen is released into the bloodstream, estrogen has thousands of targets in the body, right? But estrogen never targets every cell in the body. Are we clear?
Yes.
That's stupid. You want the signals to be specific. So one of the main functions of estrogen is to have liver make egg white protein, because estrogen has to do with ovulation. Correct? Now, do you want my estrogen to target my liver to make egg white protein? That's useless in me. I have no use for egg white protein. I don't make eggs.
Yes.
Nobody looks convinced. I am a man. Okay? I don't need to make. It's amazing how much brain damage my lab does. Okay, so understand that I should not be making egg white protein. If I'm making egg white protein, I got problems. Are we clear?
Yes.
And I got a lot of explaining to do to my wife. So no egg white protein. Why is that important? Because in women, estrogen should target the liver to make egg white protein. Should it target kidneys to make egg white protein?
No.
No. That's going to block the kidneys from working, right? You don't want the heart to make egg white protein. It has to be only the liver. Now, do you see the importance of the word target? And targets are also time dependent. For example, only in a lactating breast will prolactin stimulate milk secretion. You don't want prolactin to be stimulating milk secretion all the time. Imagine a woman who's not lactating. Okay? By the way, men have fully functional breasts. I don't know how many of you know this. Okay, that's why men get breast cancer. Men have fully functional breasts. Okay, their regulation is definitely quite different from the female. The male breasts are much more sensitive to hormones. Okay, 2 days of exposure to hormones will make the male breast make perfectly nutritious milk. Okay, men can make perfect. Not all mammals do this. Humans have evolved to make the breast fully functional. So if the baby suckled on the male nipple continuously for about 3 days, okay, and I'm talking 2 hours at a stretch, for 3 days, the male breast can completely make milk and be able to suckle. Except the problem is that system is not perfected, and it will guarantee breast cancer in men. So the system is not really up to par. Okay, we still have ways to go. We still need to evolve. But male breasts can make milk, and prolactin can stimulate the male breast to make milk. At this point, we secrete prolactin every day. Men make prolactin, women make. There's no such thing as male hormone, female hormone. Erase that from your brain. If I ever find you saying that, you're a sinner. Okay? I cannot forgive you for that. There is no such. This is where sexism begins. Okay? There is no such thing as male hormone, female hormone. Are we clear?
Yes.
You will never ever utter that. Okay? If you're the chief of nursing staff, okay, at San Francisco General 20 years from now, and you find yourself saying to a patient, male hormone, female hormone, imagine this frowny face. Okay? I'm putting the voodoo curse on you. You never ever say that. All hormones in the female are found in the male. All hormones found in the male are found in the female. Are we clear?
Yes.
Sir.
They're all common. What is different is target cells. And what is different is the effect of these hormones. In the males, prolactin stimulates spermatogenesis. Prolactin is very, very important for spermatogenesis. Prolactin is very important for kidney function in everybody, male or female, doesn't matter. In addition, prolactin causes milk synthesis in a lactating female. Do you understand?
Yes.
Okay, the breast has to receive the prolactin signal to make that milk. So the breast has to be in that condition. Not all breasts are welcoming to prolactin. Are we clear?
Yes.
So male breasts, in fact, have no receptors for prolactin. That's why we're not making milk randomly, correct? Or we'd be walking milk jugs, right? It's not going to help, right? It's a waste of energy, right? Imagine the number of calories. I mean, we don't make skim milk, right? So full fat milk, and it takes thousands of calories to make milk. Okay, it's not, the male will probably die. Right? They're not going to have enough energy to put up with all this. Okay, so the bottom line, why am I giving you all these stories? Don't go by the name of the hormone. The name of a hormone is simply how it was discovered. That's all. Okay, prolactin means prolactation, right? That's how it was discovered. Are we clear? In the rabbit. That's how it was discovered. Injecting this hormone caused rabbits to lactate. Okay, but That's not all it does. Prolactin is very important in fish. When fish move from salt water to fresh water or fresh water to salt water, right? You know, salmon travel, right? Prolactin is absolutely crucial for their body adjustment. So prolactin does all kinds of things, okay? We just think of it, we just look at it from a discovery standpoint and say prolactin supports lactation. It has many, many other functions. In a non-pregnant woman, prolactin is very important for ovulation. Okay, if you have prolactin problems, you're not gonna get pregnant in the first place. Are we clear?
Yeah.
So in the males, prolactin is extremely important for spermatogenesis. Prolactin is released right at at 3 in the morning or 4 in the morning, right around the PMS period of the male cycle. So when males go through PMS at 3:30 in the morning every day, males go through PMS every single day, that's when prolactin is released as a stress response. Is that clear?
Yes.
So don't ever think female only, male only. That never exists. So a hormone will produce very specific effects. Prolactin in the testis will promote spermatogenesis. Prolactin in the kidney promotes salt balance. Prolactin in the ovary will promote ovulation. Prolactin in the breast promotes milk synthesis. Lactating breast promotes milk synthesis. Are we clear?
Yes.
Yes.
So very, very important, very specific effects. And lastly, a hormone has to be destroyed. No hormone stays on forever unless it's plastics like BPA. How many of you have heard of BPA? Bisphenol A? Right? You look at water bottles and say BPA free, right?
Yes.
BPA is an estrogen. And it will remain in your system for freaking ever. Okay? Because the liver has no idea how to metabolize it. And nowadays, lots of papers I'm reading. I saw a paper yesterday actually, which looked at hundreds and hundreds of prostate cancer samples. Okay? 90% of them contain microplastics. The cancer contained microplastics. Okay? So just imagine the kind of pollution we're working with. Okay? So I'm not asking people to become paranoid and go crazy, right? Because you have to live where you live. You can't be in a sterile environment, right? You can't do that. But at the same time, be cautious because you never know what's harmful, right? BPA, 40 years ago, nobody thought it was harmful,
Yes.
right? And all bottles, baby bottles had BPA, right? So, but now we know BPA is a potent estrogen, potent, okay? So we don't want that. Now, the latest paper that was talking about is a compound in toothpaste which have been very silently banned. The US banned it very quietly because they didn't want to make a huge issue, and this was banned only in late 2010s. Triclosan. Triclosan is supposed to be a hormone disruptor, right? And it disrupts estrogens. It's a major estrogen disruptor, okay? And triclosan in mice caused all kinds of effects. And triclosan used to be a big part of all toothpaste. Big deal. And it was part of your detergent, right? Dishwashing fluid. Okay? So triclosan was everywhere. It was in your soap. Okay, body soap, right? Liquid body soap. Because it's an antiseptic. It kills bacteria, right? So they would use triclosan. It had ridiculous type of effects. Particularly not in the person, but if it's a woman and she's bearing a female child, it almost completely destroyed the reproductive tract of these females. Okay, the babies, the girls. Okay, very, very bad. And these women came up with all kinds of reproductive cancers even before the age of 10. Right? You do not want that. So US quietly banned it. We still make triclosan and we export it. This is how responsible we are. We're like, oh, we can give the rest of the world is immune to this, right?
They haven't complained yet.
You don't want me to start off on that. But the point is, hormones are destroyed unless they're artificial. Okay? Most hormones, insulin is destroyed within 30 seconds of it working. Insulin is destroyed. Okay? Steroid hormones take longer to destroy because they're smaller and they're fat, right? So the more fat we have in our body, the more you store your steroids. And that explains the obesity link to breast cancer and prostate cancer because they're both steroid-dependent cancers. breast cancer, estrogen, and prostate cancer, androgens. Okay, they're both fat-loving hormones. So if you have a lot of fat in the body, these hormones can be stored there. And that can directly increase your risk. Okay? But nonetheless, most natural hormones are broken and destroyed and cleared in the urine. Are we clear?
Yes.
Go through that definition. Tell me if you have any questions. I really want to know what this is. This is bothering me. Portal?
Professor,
It's not even glass, it's plastic.
Professor, what is the meaning of the prolactin specifically? Prolactin.
Prolactin is a particular hormone. It's an example.
Okay.
Okay, prolactin is, the name suggests that it promotes lactation. And I was trying to say that that's not all it does.
Okay.
So be careful with the names. Okay?
Yes.
They have other functions. Anything else?
What about the receptor sites themselves for the hormones? Are they?
We're getting into that. That's all we're talking about today is receptors. Any other questions? Everybody okay with this?
Yes.
All right. So what about local secretions? And these are really important because these never see blood. Local secretions never see blood. They become very important when we get into cardiovascular and kidneys. Okay. So these are chemicals that can be secreted locally. Locally means within the neighborhood. It doesn't really get into blood. Okay. And these modes are important. I will ask these. So if H is shorthand for hormone, okay, S is source, T is your target cell. Yeah, everybody okay with these abbreviations?
Yes.
All right. So the first mode is the source cell releases a hormone and it travels in the neighborhood. Remember, it's not traveling in blood, right? It's just traveling in the local neighborhood, right?
Yes.
In a tissue. And it affects a target, okay, here. So this is your target cell, okay? So the hormone's released. Well, technically it's not really a hormone. It's a local chemical message. Because remember, a hormone by definition has to go through blood, right?
Yes.
Yeah.
So I'm just using H for convenience sake. So whatever chemical, H, okay, will affect a neighboring cell. This is called a paracrine pathway. Paracrine, para means around. Right? Everybody's heard of the word paraphernalia, right? Around, okay?
Yes.
So it's around in the neighborhood, but not going into blood. Are we clear?
Yeah.
Yes.
So if I, okay, throw candy at somebody, right? Let's say plucky chicken. I throw candy at him. That's paracrine, right? Because he's not right next to me, right?
Yes.
Okay? So there are other people, okay, between me and him. That's paracrine. Now, a special condition is what if the target is right next door? So the source and the target are touching each other, right? This is a special type of paracrine, right? It's your next door neighbor, right? That's called juxtacrine. You've heard of the word juxtapose?
Yeah.
Juxta means right next to. Okay, so that's juxtacrine. And then the third secretion is really interesting. So again, paracrine and juxtacrine happens everywhere in the body. Okay, it's really, I don't really have particular examples. We see this all over, okay, the body. But remember, they're not seeing blood. That's the main point. Only endocrine sees blood, right?
Yeah.
These secretions don't see blood. Most of these, what we call growth factors, usually. Okay. So autocrine is really interesting. Auto means self, right? So what happens when when the source and the target are the same cell? Source and the target are the exact same cell. This happens a lot with immune cells, and we're going to go over this the last 2 weeks of class, where we talk about autocrine signaling. Autocrine signaling is really important, and cancers actually develop inappropriate autocrine signaling. There are companies in South San Francisco dedicated to just studying this. Okay, multi-million-dollar cancer drug companies that do this. Okay, so autocrine is self-stimulating. Self-stimulating. 4 is a very interesting, very rare pathway. This doesn't really happen much. Okay, and that's called intracrine. Intra means within, correct?
Mm-hmm.
So intracrine means within. So the hormone is released inside the cell. It's not even getting out of the cell, right? The receptor's right there. Lot of androgens do this, testosterone, okay? Lot of androgens are intracrine, very intracrine. In fact, cells will convert them to estrogen within the same cell. Okay, so that's an intracrine pathway. And you already know neurocrine. And you look at that deformed neuron, right? You've seen this before, right?
Yes.
Action potentials come down, calcium comes in, and you release neurotransmitters, correct?
Yes.
Neurotransmitters, please say yes.
Yes.
Okay, so that's neurocrine. Okay, so that's the 5th pathway. And neurons do release hormones. Neurons do release hormones, but here we're not talking about hormones. We're talking about neurotransmitters. Remember, this is all local modes, correct?
Yes.
They do not get into blood. Neurohormones do get into blood, right? There's a difference. So if it makes you, if H is very confusing, replace H by C for chemical, okay, if you want. Or you can say LC, or local chemical. Okay, so if you want to do that, I can't erase this. So if you want to replace H with that, that's fine. Hormones, by definition, have to go through blood. There's no exception there.
Why would a cell do autocrine instead of intracrine?
There is no why, it's just, you have to look at the physiological context. So for example, immune cells,
Yes.
most immune cells are inactive.
Right.
So when they come in contact with their target,
Mm-hmm.
right, they need to make lots of copies of themselves. So that's an example of a local stimulation.
So it's not just some cells, it's also a paracrine of sorts?
So autocrine and juxtacrine are subsets of paracrine. Okay, and autocrine is really special because the source and the target are one and the same.
Okay, I see.
Autocrine means self-secreting, self-stimulating, right?
Yeah.
Whatever you secrete, it's affecting you. Make sense?
Yes.
Yes, sir.
For intracrine, did you say that the target cells are also inside the cell?
There is no, so first of all, the chemical is not even secreted. So it's really an anomaly, right? Because crine means secreting, right? So the hormone doesn't even get out of the cell. The receptor is already inside. So the hormone is made, it binds the receptor in the cytoplasm, and you have an effect. So the hormone doesn't even get out. Okay? That's extremely rare. Okay? You don't really see that. It's very, very uncommon. So 4 is very uncommon. A lot of people haven't even heard of intracrine. Okay? Again, you're getting all this because I was taught by fathers of endocrinology. Okay? So, and they coined all these terms.
So fascicles within the thyroid would be autocrine?
Colloid. Follicles.
I mean, so, yeah, colloid, sorry.
Follicles within the colloid.
Sorry, yeah. The cells would be autocrine then, correct? Because you're secreting the,
You're assuming that the colloid, the hormones from the colloid,
yeah,
are signaling into these cells.
yeah.
That's not, how can you make that assumption? I don't know.
Oh.
If they did, then yes. But I don't think they do. Okay? So anyway, don't worry about that question. Not until Thursday. All right. So local secretions, they don't get secreted into blood. Growth factors undergo local secretions, right? So signal, right? So let's say, 2 rows down, okay, Plucky Chicken's injured. I want to repair Plucky Chicken, right? So then I will send a growth factor to him to repair and get rid of the waste and things like that. Are we clear?
Yes.
Many chemicals in the immune system use paracrine secretion, okay, to help immune cells navigate through the tissue, okay, to find the bacteria, right? So it's all examples of paracrine signaling. Are we clear?
Yes.
So again, anything that's not going into blood could be paracrine, juxtacrine, autocrine, or intracrine. Clear? Neurocrine, neurocrine is all neurotransmitters. So that's we've already done that one before. I'm drinking water to slow me down. Any questions on this? Thursday section, were you able to find these notes?
Yes.
Okay. All right. So I think most of you are here. So I'm going to do the vote, and then we'll decide, and then we'll move on to the second part. So the proposition is, okay, that next week you'll have your exam. It'll be a 50-point exam only on muscles. Endocrine is not included because there's a lot of material to master, and this is very important material. I don't want you rushing through this, okay? So no endocrine on next week's exam. The week after, you don't have any exams scheduled. So in other words, I'm splitting the 100-point exam into two 50-point exams.
Yes.
Okay, so next week you will take the muscle exam, and then the week after you will take the endocrine exam. Yes?
Yes.
How many of you are okay with this? I need show of hands. Okay. Use late. What?
I don't know. I'm working a lot next week, so I'm just thinking.
Well, you took 103 A and B. That's not
Okay.
fair. I'm not going to take your vote. So, how many of you cannot do exam 2 weeks from now? Talk to me. We'll arrange something. Okay. So, but yeah, I'm not putting this endocrine on them for next week. That's absolutely cruel and unusual punishment. So, and I've already written the exam, so it doesn't affect me, but you're not going to be able to handle this. You won't learn anything. You're just going to memorize and everything's going to fall flat on your face. That's not good. So I want you to just take muscle next week. So I'm confirming it with you right now. So next week, the exam is still on, okay, during lab. It'll be a 50-point exam, okay, and only for an hour, okay, not an hour and a half. It'll be a 50-point exam, and then you'll have the second endocrine exam the week after. Everybody clear about this?
Yes.
I'll put this in announcements.
Yes, sir.
Okay, so classification of hormones. So hormones can be classified in many, many, many ways, okay, and it's a little booklet in itself how hormones are classified, but I'm going to go through only one classification, which is important. And it's based on solubility in water, okay, it's based on water solubility. So if you're hydrophobic, you can be grouped into one category, and those are fat-soluble hormones. Fat-soluble hormones, those are hydrophobic hormones. And the second category is water-soluble hormones. Hormones that are soluble in water. So fat-soluble hormones include cholesterol and all its children and grandchildren. Okay? Cholesterol, in some animals, is known to have hormonal effects. Okay? Not in humans. Okay, so cholesterol is not a hormone in humans, but cholesterol is the grandmother of all steroid hormones. Cholesterol is the starting molecule, okay? So cholesterol and its steroid derivatives, right? And fat-soluble vitamins, and that's vitamin A and vitamin D. Those are considered steroid hormones because we have the receptors for them. We know what the receptors are. So vitamins A and D, there are two more fat-soluble vitamins, E and K. They're not hormones, are we clear? Because we don't have receptors for them. These two have receptors, the retinoic acid receptor and the vitamin D receptor. Okay, and one of my very close friends, she cloned the vitamin D receptor in 1987. So she discovered that. So that's a big deal. Okay, vitamin D. Thyroid hormones. Thyroid hormones are very hydrophobic. They're very hydrophobic, meaning they love fat. They don't like water. Are we clear? You should know this by now from bio. So thyroid hormones. So these are all fat-soluble hormones. Now, T3, which is triiodothyronine. Tri means three, right?
Yes.
Iodine, thyrronine, okay, and tetraiodothyronine. 4 iodines, right? Tetra means 4. So tetraiodothyronine, T4. So these two together are called thyroid hormone. So whenever we say thyroid hormone, it's referring to T3 and T4. Are we clear?
Yes.
Sir.
Okay. And prostaglandins, prostacyclins, leukotrienes, those are all local fat-soluble hormones. Local, meaning do they see blood?
No.
Oh.
No. They're usually paracrine. Okay. Why are they called prostaglandins? Because it was discovered in the rabbit prostate. It was discovered in the semen of rabbits. Okay. So they're prostate secretions. So they're prostaglandins. Prostacyclins are the same way, right? So prostaglandins and prostacyclins, why are they present in the semen? Because that will make the uterine cervix contract, okay, smooth muscle contraction, right? And that will lead the female to orgasm. Are we clear?
Yes.
So it's very, very important for fertility. So those were all discovered with rabbit semen. And leukotrienes are very important in injury. So anytime you have a cut or some kind of injury and you feel that burning sensation, that's all due to leukotrienes. Okay? We will discuss all of these throughout the semester. Okay, we're going to be talking about prostaglandins and leukotrienes all over the place. I'm not going to talk about them now. Just know that they're local secretions. Are we clear?
Yes.
Is the list of fat-soluble hormones okay? Fat-soluble hormones list is okay?
Mhm.
Okay. Water-soluble hormones is very easy. Everything else is water-soluble. Okay? Huge list. Okay? All the peptides and epinephrine. And you already know epinephrine, right? Sympathetic nervous system.
Yes.
Hello, last unit.
Mhm.
So adrenaline, right? And sympathetic nervous system. Epinephrine is a water-soluble hormone. Thank heavens, right? So interestingly, both epinephrine and thyroid hormone are derived from the same precursor, from tyrosine, which is an amino acid. And sometimes they're both called amino hormones. Okay? But I will not be using that term. Are we clear?
Yes.
Anything with a star, I will not be using. So don't worry about it, but I just wanted you to know, because sometimes some old books will have that, right? And some people who follow old terminology, old school terminology, use that, right? They call it amino hormones. So adrenaline and epinephrine are the same thing. You guys should know this by now. So any questions on fat-soluble, water-soluble list? You need to know this table. And when I say no, I mean no. Okay? Not memorize and vomit. I need you to know the tables. I need you to know the concepts, because these are very, very important concepts that you're going to be using throughout the class. Everything in our body is coordinated by neurons and hormones. Are we clear?
Mhm.
So you need to know this. This is really foundational. So chemical nature, fat soluble, obviously they're hydrophobic, right? If you're going to dissolve in fat, you're going to be hydrophobic. Anybody have any qualms on that? Hydrophobic means scared of water, right?
Yes.
Fat is scared of water. Water-soluble hormones, however, are amphipathic. Amphipathic means they have water-soluble parts and water-insoluble parts, like your membrane. Correct? Amphipathic, it's both. So where in the cell are these made? And this is important. Where in the cell are these made? Fat-soluble hormones, remember all lipids, this is Bio 102, all lipids are made in the smoothie R. All lipids are made in the smoothie R, okay, from 102 days. So no exception, fat-soluble hormones are made in the smoothie R. And you know smoothie R to also do what? What did we talk about smoothie R a lot about? Last week, what did we talk about it? Sarcoplasmic reticulum, right? That's smoothie R, right? Water-soluble hormones are secretory, so they're made by rough ER, okay? That's the endoplasmic reticulum with ribosomes. Storage at source is impossible. I've already explained that. They're hydrophobic. They go right through the membrane. They simple diffuse through membranes. So anything that simple diffuses through the membrane, you can't store it. Can you store oxygen?
No.
No. Can you store carbon dioxide? No, because they go right through the membrane. Are we clear?
Yes.
Can't store them. And for water soluble, you do always. You keep them in reserve. right? You store them in vesicles. Right? And they're kept as a reserve because most water-soluble hormones control life-threatening variables. They're responsible for life-threatening variables. Example, blood pressure. Okay? How do they travel in blood? Remember, fat-soluble hormones don't like water. That's why they're hydrophobic. Phobia means fear, right? So how do they travel? You have special proteins on which you travel. Less. So they travel on specific proteins. Remember, proteins are always specific, right?
Yes.
So those proteins are made by the liver, by the way, which is why when someone has liver failure, okay, particularly in women, their cycles go haywire, okay, because the estrogen that they make and the progesterone they make, they don't have these proteins to piggyback on, right?
Yeah.
So the brain starts thinking that the woman's pregnant because the levels are high, right? Because they don't have proteins to grab and sequester them, right? So the brain senses a lot of estrogen and progesterone, right? And goes into the assumption that the woman's pregnant, and then she stops ovulating. Make sense? So which explains why she doesn't go through her periods properly. So liver failure is a big deal, right? So all these proteins are made by the liver, okay? They're called transport proteins. They carry specific steroid hormones, okay? There's a thyroid binding globulin. There is a sex hormone binding globulin. There is a corticoid binding globulin. So you have very specific transport buses that carry specific hormones. Are we clear? Do you need to know all of those? No. I'm not going to require you to know them, okay? But very, very specific, okay? And for water soluble, they don't need any help. Blood is all water. They love water. They don't have a problem. Pause, and I'll ask if you have questions on this so far. I would sit down and recopy this whole damn thing into another piece of paper. That's the best way to study for this chapter. Just blindly copy it. Okay? I promise you, you'll understand this a lot better when you do that than sitting and staring at the notes. Sitting and staring is the worst thing you can do. Okay? Either teach it to your friends, or teach it to your dog, right? Or I remember when Shiva used to be a baby, I used to tie him up and I used to practice all my lectures on him, and he'd fall asleep in 5 seconds. It was perfect. And when he went for his kindergarten interview, there was an interview, believe it or not, at Challenger. So he went for his interview, and they asked him for a word that started with a Q, right? And he said, quadriceps femoris. They admitted him on the spot. He retained it while he was sleeping, because I was practicing all my lectures on him, right? And my wife was like, what did you do to him? Anyway, so teach it to others. It's really a good way of learning this. Number one, if you don't want to do any of that, you're antisocial, you don't like people, okay? I know how you feel. And I support you, that's fine. Sit down and copy this into another piece of paper. It'll take you half an hour, and it'll save you 8 to 10 hours worth of work. Just recopy it, word by word, into another piece of paper. It is so worth it. Okay. Receptor site. So where do you find the receptors? True or false? Receptors are very non-specific.
False.
Okay? Receptors are very specific, okay? And receptors are going to be inside the cell for steroids. Why? What can steroids do?
Oh,
They cross the membrane by diffusion, correct? So the
Yes.
receptors are going to be inside the cell. Okay? And what about water-soluble hormones? They're big proteins. Can big proteins cross the cell membrane?
No.
No. So where would you find their receptors?
On
On the membrane of the cell, plasma membrane. Are we clear?
Yes.
So they're on the plasma membrane. Mode of action, this one's extremely important because this is really the crux of today's class. Fat-soluble hormones are slow as heck, okay? They're very, very slow. One very simple example, puberty, okay? Puberty goes on and on and on and on, right? Starts at whatever, 10 or 11, 12. Nowadays, the average is 9, right? Starts whenever, and then goes on and on and on, okay? I literally just developed knuckle hair, okay? I mean, seriously, in the past 2 years, I'm like, what is this, right? Like, it never ends, okay? So, and it's true, puberty never ends. It keeps on going. The effects are so permanent, right? As soon as your vocal cords thicken in males, that's it, it's permanent. It's not going back to your child voice, right? Once a woman goes through pregnancy, the changes in her reproductive system and the changes in her brain, permanent. No going back. Are we clear?
Yes.
So, very important. So, fat-soluble hormones are very slow, is the idea. Now I'm going to explain to you why they're slow. Because what they do is they modulate transcription of target genes. Please tell me they did this in Bio 102. You've heard of the word transcription.
Yes.
Okay? What is transcription? You make RNA from DNA based on DNA as a template. You make RNA and use that RNA to make protein, right? This is part of gene expression. So if you don't know or you've forgotten this, please look up gene expression on YouTube and watch a 1 to 2 minute video. Very important, but that's what fat-soluble hormones do. Fat-soluble hormone receptors are transcription factors. They're actually transcription, they're ligand-gated transcription factors, meaning the hormone turns them on. And they go look around the DNA and turn on specific or non-specific genes. Very specific genes. You do not want testosterone turning on egg white synthesis. Are we clear?
Yes.
Major confusion.
So is that why, when you said earlier, estrogen and all those things cause cancer? Like
That's why we think they cause cancer. I mean, estrogen causing cancer, I have a lot of problem with that. Okay, with that theory. But who the heck's going to listen to me? Okay. Despite proving it. So the scientific community doesn't want to look at it at all. Till today, everybody's screaming estrogen, estrogen, estrogen. Okay, when I'm telling you it's not estrogen, you're barking up the wrong tree. That's why breast cancer is not even 10% curable. Okay, because you're completely barking up the wrong hormone. Okay, anyway, that's what fat-soluble hormones do. They modulate expression of genes. Is that going to think about it? The receptor has to go in. We have 35,000 genes, right? You have to go and find your set of genes you want to turn on.
Yes.
And then you have to transcribe. Then take the RNA and you have to modify the RNA. And then that RNA goes into the cytoplasm. And then ribosomes start cranking out the protein. All this is going to happen in 2 seconds?
No.
No, it's going to take its sweet time. Some steroid responses take years to achieve. Okay, I'm not kidding. Some steroid responses take years to achieve. Okay, that's why puberty is not completed in 2 months. Am I making sense?
Yes.
It takes forever. Okay, so de novo protein synthesis. So here's the Queen of England. De novo means from scratch. De novo means from scratch. If your blood pressure is falling, do you have this kind of luxury? To go into genes, find the right genes, make mRNA, modify mRNA, take that mRNA, make protein, and then have an effect? You'll be long dead and gone, correct?
Yes.
For blood pressure drops, you want the response to be on the spot, within seconds, correct?
Yes.
So that's why water-soluble hormones, they only work on pre-made proteins, proteins that have already been made. Okay?
Yes.
Tell me, if a room is dark, what do you want to do? And let's say there's somebody with a knife, right? Let's make this more fun, right? Somebody hiding, I know what you did last summer type nonsense, right? And you hear my voice. I know you didn't study. And you want to turn the light on right away, right?
Yes.
Do you want to go make the bulb and then fit the bulb and then make the circuitry, right? And then turn on the bulb?
No.
Okay, so think, common sense. Water-soluble hormones have to work instantly or you'll be dead. Are we clear?
Yes.
It's that simple.
Yeah.
Okay? So they will involve signal, your favorite word, signal transduction. And you know what signal transduction is, right?
Very
I
slow.
turn John on, and then John turns to Cami, activates him, I should say activates Cami activates Plucky Chicken, and this keeps on going. So how long does it take? If I pass on the stick to John, John throws it to Cami to Plucky, right? Boom, it's done, right? It's immediate. Am I making sense? You add phosphate groups, okay, and it happens immediately. Okay, signal transduction is rapid. Okay, so water-soluble hormones need rapid responses. Glucose uptake takes less than a minute. Less than a minute. Because when your cells want glucose, they want glucose, otherwise they're going to die. Right? There's no option there. So, more bougie terms, transience of effects. Okay? What is the regular term for transience? Temporariness. Right? That's not a word. But I consider the ghetto version of transience. Temporariness. Okay? How temporary is the effect? Right? Is it long-lived or is it short-lived? Well, fat-soluble hormones, obvious. It's permanent. Right? You're going through all this effort, so the effect only lasts you 2 minutes? No. Puberty lasts forever. Pregnancy lasts forever, right? The effects. So, permanent effects. Whereas insulin, do you want insulin to be a permanent effect?
No.
You'll die of hypoglycemia, right? All the glucose gets taken into blood and that's it. Story over, right? So those effects need to be very, very transient. Transient means only last for a few seconds. Complete opposites. Okay? Having done this explanation, this is obvious. Fat-soluble hormones are super slow in how they act, right?
Yes.
Because they're changing things like reproductive ability, right? Pregnancy, right? The last thing you want is for these hormones to be hasty and quick, right? You have to do it carefully. So slow. When a cell is starving, its glucose needs are immediate. It's not going to wait. It's going to die if you don't give it enough glucose. Lastly, and surprisingly, and again, I don't really know this. I keep wanting to do this research, and I've been saying this for 12 years, and I keep forgetting. So maybe y'all can do this and tell me. Usually, diabetics don't have high rates of cancer. Okay, if you think about it, if someone's diabetic for a long time, okay, you'll notice that those cancers don't grow. They have cancer. Sure, that everybody's born with 6 cancers. Okay, shameless advertisement. Take my cancer class in this one. Easy class. Never have to see my face once. So that's got to mean something. Anyway, but diabetics don't have high incidence of cancer, right? Most probably because diabetics can't take in sugar. And if cancer cells can't take in sugar, they're all going to die. Am I making sense? It's like sickle cell anemia being advantageous to malaria people, right? People who have sickle cell, their life sucks. But if they live in Africa, they're protected from malaria, right? So there is some blessing there of some sort, right? Diabetes, horrible disease, right?
Yes.
Okay, but you do have some kind of a protection. But I don't know if that's actually causally true. I have to check that out. I actually don't know. I've been always wanting to look at that data. But I do know, as personal anecdotes, that a lot of diabetes patients don't usually report cancer, maybe because they die early from diabetes. They don't live long enough to get cancer. Make sense? But I cannot but imagine that not being able to take up sugar is going to be a big disadvantage for cancer, given its metabolic needs, right?
Yes.
So, and also blood not being able to flow. very comfortably, right?
Yes.
Cancer needs that blood vessel supply and blood supply and oxygen, right?
Mhm.
So something is good about having diabetes. Okay. Last, half-life. Half-life is how long the hormones last you. Now, fat-soluble, I told you, right? They last forever because they're tiny. The liver has a hard time finding them and breaking them down. This is why microplastics stay in our body forever, because liver can't find them. They're obvious, you can get rid of them, right? They're teeny tiny targets, how are you going to find them? So fat-soluble hormones are extremely small in size. They're very, very small. The liver has a hard time finding them, okay? On the other hand, big proteins, you can find them, right? And easy to chop up, right?
Yes.
And in fact, blood has a lot of enzymes that break down these hormones. For example, insulin. Insulin can be broken down very easily. Okay? I want you to look at this table, catch up with your notes, and ask me questions. Everybody following along with this? Any questions? I got a question on this on the N class of all things. It's really weird, but steroid receptors, okay, showing up on the N class. Okay, very interesting question. How do hormone receptors work? So what do they do? How do they work? Okay, so I'm giving you all these details, but I'm not going to test you on them. Okay, so you can actually just I'll tell you what I need you to know. There's more detail here than what I need you to know. Okay, so I'll tell you what you should focus on. Steroid hormones diffuse through the cells by simple diffusion. Everybody got that? Yes?
Yes.
We talked about it, right? They bind one receptor. When they bind one receptor, and receptors are specific, right? So estrogen receptor will not bind androgens. Are we clear?
Mm-hmm.
So estrogen receptor is going to bind estrogens, right? So any steroid, right? Corticoids, right? They bind, cortisol will bind its receptor. Aldosterone will bind its receptor. So they're all specific receptors. So you take one receptor, okay, and watch my hands, right? So this is a receptor, right? So when the hormone binds, I grab onto it, correct?
Mm-hmm.
So my shape changes, right? And then I go around looking for another partner with the same idea, right? Where the hormone's bound,
Mm-hmm.
right? These two come together. This is called dimerization. Di means two, mer means unit. So dimer means 2 units. So 2 receptors come together and they activate each other. Are we clear?
Yes.
So this unit now travels to the nucleus and, okay, very interesting. You don't need to know this part. When these 2 receptors bind, yes? Watch my hands. When these 2 receptors bind, okay, these fingers pop out. And literally that's what they're called. They're called zinc fingers. Okay? So when 2 receptors dimerize, you have a change of shape, correct?
Yes.
And these fingers go in search of their DNA, the specific genes, right? And then they grab on them, and then you have transcription. Okay? So what do I need you to know for the exam? Steroid receptors are found in the cytoplasm. Steroids diffuse in, okay, and turn on the steroid receptors. and you produce new protein. That's all I need you to know. Are we clear? I
Yes.
don't need you to know zinc. I don't need it. So now think about it. If you're zinc deficient, you're going to have big problems. Okay? Anyone wondering, the whole, what is it, the multivitamin commercial, A to zinc, Centrum, right? It says, or is it too, are you all too young to have heard of Centrum? A to zinc, right? The zinc is really, really important in your multivitamin, okay?
Yes.
Because if you're deficient in zinc, you're not going to be able to do half these things, okay? So zinc is very important in transcription, okay? Otherwise, you will not be able to transcribe, okay? So transcription factors use zinc, okay? And the idea is steroid hormones activate their receptors, which are found inside cells, and these will turn on specific genes, specific gene expression, right? And that allows you to make brand new protein. Are we clear?
Yes.
So it's a huge energy commitment. Okay? You don't need to know any of this co-regulator nonsense. Okay? Don't worry about it. I used to teach this in much more detail, but it's not necessary. Okay? Interesting fact, again, I will not test you on it, but these steroid receptors can be inappropriately activated by growth factors. Now, normally, these receptors should only be activated by their respective hormones, correct?
Mm-hmm.
But they can be activated inappropriately by growth factors. So if you turn on androgen receptor, okay, because you have these growth factors that are around, and androgen receptors turn on without the androgen, okay, it's very difficult to turn them off. Are we clear?
Mm-hmm.
Because the body's not used to this, right? And androgen receptor will make the prostate cells divide, and that can lead to cancer.
Mm.
Am I making sense? One little mistake. That's a mistake. It's not natural. It's a mistake. Okay? So you don't need to know that for the exam. Okay? But just what I told you. How do steroid hormones work? They turn on specific genes and make new protein. That's how they work. And it's slow. Right? Everybody got it?
Yes.
Water-soluble hormones, they cannot enter cells because they're large and polar. They're big proteins, right? So they will cause signal transduction, and you've already seen signal transduction before, right? Indirect neurotransmitters, how do they work? They bind the receptors, right? The receptors turn on G proteins. G proteins will activate cyclic AMP. Cyclic AMP activates calmodulin. Remember all this smooth muscle contraction?
Yeah.
That's signal transduction. That's how protein hormones work, okay? Most of the time, the hormone and the receptor are endocytosed. You should know this by now. They're endocytosed. That's phagocytosis, right? They're endocytosed and destroyed by the cell. Are we clear?
Yes.
If they're not endocytosed, you're in deep trouble because the signal is now never off. Right? A normal cell does what? Takes in the receptor and the hormone and destroys it. Destroys both of them, right? Usually the receptor is recycled. The hormone's destroyed, okay? But if you're not able to endocytose, then what's going to be the problem? The signal will always be on. There's no way it's turning off, right?
Yes.
That's again an issue. Now, with water-soluble hormones, there are two mechanisms. One of them you've already seen, the indirect neurotransmitter mechanism. So there are two mechanisms. One is called amplified mechanism, and the other is called a non-amplified mechanism. Okay? The non-amplified mechanism has a very interesting name. It's called tyrosine kinase mechanism. Tyrosine kinase mechanism. And the amplified mechanism is called second messenger mechanism. Second messenger. And just to give you a definition, phosphorylation means addition of a phosphate. Acetylation means addition of an acetate. Methylation means addition of a methyl group, right? So anything Asian means you're adding it. Yes?
Yes.
Sumoylation is the addition of a sumoyl group. Okay, and that is a word, okay? The adding of the phosphate will cause conformational change, which can then turn on or off a given protein. Okay, it can be either one. So when you look at this pathway, if you look at this mechanism, okay, GF stands for growth factor, RTK stands for receptor, tyrosine kinase. The growth factor binds. Now notice that's an integral membrane protein, right?
Yes.
That turns on, and it turns on GRAB2. Now, do you need to know any of these? No. Please, God, no. Okay? I don't even make my fourth year undergrads know these names. Okay? Usually grad students, I'll require them to know this pathway. Okay? You don't need to know any of these names. Are you clear?
Yes.
What is the point? So GRAB2, GRB2, okay, is what type of protein? Peripheral or integral? Peripheral, right? It's a peripheral protein. And remember, it's loosely attached, correct?
Yes.
Because it carries the message through. Do you see the relay there? GRAB2 activates SOS. SOS is son of sevenless. No, that's literally what it stands for. And son of sevenless will activate RAS. RAS is rat sarcoma. Okay? RAS will activate RAF, fibroadenoma. That's the name of the protein. And then RAF will activate MEK. Okay? And MEK will activate ERK. Okay? And they all have expansions, big long expansions. And ERK will go into the nucleus and turn on any protein making that needs to happen. Now understand that this is not a rapid effect, right? This obviously is slow. Now what can happen is MEK, okay? This is a very specific pathway. It's a growth factor, right?
Mm-hmm.
That means you're making the cell divide, right? To make a cell divide, that's going to take time. It's not something that happens like that, right?
Yeah.
You will see a very similar pathway for insulin, right? Except it's going to be a rapid pathway, and it's going to cause glucose channels to go into the membrane. Are we clear?
Mm-hmm.
Pretty rapid, right?
Yes.
But when you're talking about growth cannot be rapid. That's not a good idea, right? Because that can lead to cancer. You don't want that. You want to be very careful about when you're trying to divide a cell, right?
Yes.
So this is a non-amplified mechanism. So why is it non-amplified? Notice one hormone, which is the growth factor, one receptor, yes?
Mm-hmm.
Anywhere here, do you see a multiplication?
No.
No, it's all 1 to 1 to 1 to 1 to 1. Do you see that?
Yes.
Okay, it's not like I am activating all 6 of you, correct?
Mm-hmm.
That would be and the 6 of you is activating 12 more, right? That would be an amplified mechanism. Are we clear? I mean,
Yes.
making it bigger and bigger and bigger, right? Here, it's a 1 to 1 to 1 relationship, and that's what I wrote 1 to 1 there. So it's a 1 to 1 relationship. So that's called a non-amplified pathway. Insulin is the best example for non-amplified. Insulin. So very easy. Insulin binds its receptor. The membrane folds. The receptor activates. And then you have proteins that are in the middle, right? And then eventually, that will cause glucose channels to go into the membrane. Are we clear? It's pretty quick. Okay? It takes 10 to 15 seconds for this to work. Because it's a domino effect, right? I activate you, you activate them, and then boom, glucose channels recruited. Are we clear?
Yeah.
So it's
Yes.
pretty rapid. So what about the amplified signal? And again, this is something you've seen before. Smooth muscle. This is what happens in smooth muscle. Okay? You're dealing with a second messenger. Now, when I say second messenger, there's got to be a first messenger, right?
Yes.
The first messenger is the hormone, correct? Because, so do you see number 1 there? Messenger binds the receptor, correct?
Yes.
And then the receptor is very interesting. It has the shape of a snake. And all these receptors do, okay? They're all shaped like snakes, right? Except the head of the snake binds a specific hormone, right?
Yes.
So specificity is at the head of the snake. So it's called a serpentine receptor, and the G protein is attached to the serpentine receptor. This is from a textbook, okay? So you have the serpentine receptor and you have these G proteins. And the G protein, so when the receptor is activated, right, it's going to kick the G protein out. And the G protein will activate an enzyme. Is this integral or is this peripheral?
Peripheral.
Peripheral, right? But it's only on one side of the membrane, right? So this is adenylate cyclase. Adenylate cyclase will produce cyclic AMP. Now here's the interesting part, and this is important because you're going to see this over and over later. I want you to imagine this, so stop writing and watch me, right? So I'm the receptor, correct?
Mm-hmm.
So I'm the little snake here, right? Everybody see my head and the tail?
Yes.
Mm-hmm. Yeah, actually I was born the year of the snake, so why not? So I bind hormone, right? I start getting activated, right? And my response is I'm going to kick the G protein out, correct?
Mm-hmm.
So if I kick the G protein, right? So say it goes to Tran, right? The G protein goes to her. She's the enzyme. She will convert one ATP to a cyclic AMP, correct?
Yes.
And she will kick back the G protein to me. Does this make sense?
Yes.
And if the hormone is still bound, I'm still kicking. So she will continue to make cyclic AMP until this hormone is gone. Are we clear?
Yes.
So as long as the hormone is bound to the receptor, cyclic AMP continues to get made. Cyclic AMP is your second messenger. The hormone being your first messenger. Okay? So as long as the first messenger is there, the second messenger continues to be made. So technically, if it stays long enough, one first messenger can make 10 second messengers, 20 second messengers, up to 1,000,000 second messengers. During labor, it's about 1,000,000. Okay, 1,000,000 cyclic AMPs are made. Make sense?
Yes.
So you see how this is amplified?
Mm-hmm.
Okay? And what do the second messengers do? Second messengers will activate a variety of signal transduction pathways. Okay? And you want this kind of an amplified mechanism, okay, particularly when you want a sustained response. What does sustained mean? Long term, correct? You want it to last a long time, right? What are some examples? Labor. Do you want labor to stop in the middle?
Nope.
No, that's a long time thing, right? You want to keep on sustaining the contractions until the baby is born.
Mhm.
And response to low blood sugar is also sustained. If your blood sugar goes low, dangerously low, okay, believe me, your heart's going to start pounding and you'll start sweating. Those are all sympathetic responses. Are we clear?
Yes.
And sympathetic system is an amplified mechanism. Makes sense, right? You just turned on the alarm. You want to be careful because you have no idea if the lion's gone or it's lurking around, right? So that's why when you wake up from a bad nightmare, you cannot go back to sleep. Because the brain's going, you want to sleep, you fool? You have something chasing after you, right? You can't go to sleep, right? So amplified responses, very important, okay? It involves second messengers, cyclic AMP, cyclic GMP. These are all second messengers. I need you to know that. You're going to use this forever after this. So you need to know it. So make sure you understand this pathway. And then you have a variety of responses in a cell. Okay? So questions on the amplified mechanism and the non-amplified mechanism. Take a look at that. Tell me if you have any questions. Yes, sir.
So for the second messenger, when the initial receptor takes the GDP over, is it GDP the messenger or is this?
No, so I don't need you to know the particular details. You don't need to, but the G protein, right? GDP and GTP are the on and off switches for the G protein. That's why it's called a G protein. A G protein is turned on by GTP. And when the GTP is used up, the GDP turns it off. Okay, it's a very weird system. Okay? So that's what that's describing, right? So if you have GDP, the G protein is off. If you have GTP, the G protein is on.
So
The second messenger is your cyclic AMP. This is your second messenger. So this is, see, second messenger has to be made in millions, right? You cannot make G protein in millions. That's a lot of, that's a protein. You can't make that in millions. Cyclic AMP is just a nucleotide, right? You can make millions of those, no problem.
So is the key feature that you're just producing multiple secondary messengers from?
Yeah, the G protein keeps recycling between me and the adenyl cyclase.
Okay.
And as long as the hormone is bound, I keep pushing that ball back to the enzyme. And the enzyme keeps on making cyclic AMP, but every ATP it's using, it wants to make sure the hormone is still there. Should I still be wasting ATP? Right? Because if the hormone is not there and the enzyme is on, right, that's bad news, right? You're going to be using up all the ATP in the cell and making cyclic AMP for no reason. Correct?
Yes.
Is that making sense, everyone?
Yes.
This is a complicated mechanism, yeah. But cyclic AMP or the cyclic nucleotide is the second messenger. Calcium is also a second messenger. Okay, it's the only ion that actually is a second messenger. Okay, calcium ions are accepted as second messengers inside a cell. So the amplification gives you a very long response, correct?
Yes.
That's the idea, it's time dependent. Okay? Whereas an insulin response is what? 1 minute and you're done. You take in whatever glucose you want, you're done. You shut off the whole thing. Because if insulin were to be amplified, you are doomed, right? Because glucose channels are going to be permanently in the membrane, taking blood sugar in. Right? That's not good, right? Your blood sugar is going to go way down, right? We don't want that. Okay, you don't need to know this. Okay, this is that steroid growth factor interaction. You don't need to know this, so don't worry about it. You can look through it if you're interested, but it's important for cancer. So if you don't want to know this, don't worry about it. So I do want to give you some more definitions, and these are mechanisms of hormone action. And we're talking about multiple hormones working together. The first is called an agonist or an agonism. So when one hormone mimics another hormone, or when two hormones are producing the same effect, when two hormones produce the same effect, okay, that's called agonism. And an example I used here is when you look at the active ingredient of soy, it's genistein, right? Genistein can act as an estrogen in the breast, okay? And this is why excessive soy can increase the risk of breast cancer, okay? Because genistein is an estrogen in the breast, right? So we call this an estrogen agonist, okay? It behaves like an estrogen. So if something acts like a hormone, that's called an agonist. Agonist means you're acting like a hormone. And the same goes for two hormones. So when two hormones, for example, I'll give you a realistic example. Progesterone can work like a mineralocorticoid in the body. Okay, progesterone can act like a mineral. So in other words, progesterone can also retain sodium. And mineralocorticoid can also retain sodium. So those would be called agonists of each other. Are we clear? They're similar in how they work. Am I making sense? They produce similar effects. An antagonist is super easy to understand, right? They oppose each other. Right? When two hormones oppose, simplest example, insulin brings down blood sugar, glucagon raises blood sugar. Those would be antagonists. Do you agree? They antagonize each other. Okay? And the example I'm using here, tamoxifen is an estrogen antagonist. Tamoxifen is an estrogen antagonist. It's an anti-estrogen, at least when it was invented. It's an artificial chemical drug. Okay, but my work, which is all gone into the wind, and tamoxifen is a $5 pill. Each pill is $5. This is what they give breast cancer patients. My work showed that tamoxifen is an anti-estrogen in a woman who's cycling, right? Premenopausal women, when the ovaries are working, tamoxifen is an anti-estrogen. In a postmenopausal women, which is what most breast cancer happens, are postmenopausal women, tamoxifen is a full estrogen agonist. The minute I found this information and I presented it at a seminar, okay, my boss gave me 7 weeks off, and I was so happy. He gave me an A+ for research. Usually you never get that. And that was Nobel Prize winning work, because it would have completely changed how we think about estrogen, right? The company that makes tamoxifen came in, robbed the lab, burned all my notebooks. 3 years worth of work gone down the drain. And I know it's them, because who else would want this data? You're not going to go rob my notebooks, right? Makes no sense. And when I came back, my professor said, you're going to have to start on something else, because we're not allowed to work on this. Guess who lost? All the women that still take tamoxifen to this day. So in these women, no wonder 84% of them come back with breast cancer, because tamoxifen is a full estrogen in a postmenopausal woman. When this drug was developed, they used rats, which is a very bad model, because rats become hyperfertile as they age. In rat populations, great-grandma is the sexiest woman in the colony. Do you understand? Not young chicks. They don't care for the young chicks. They want the oldest, most experienced female. Are we clear? Because she has the most estrogen. And that is not how humans work. Humans, after a certain age, we lose all the estrogen, right? So this hormone has a completely different effect. Okay? So when you put things into your body, you have no idea what it's going to do. Okay? And so all I did was I wanted my model to resemble postmenopausal women, because that's where 80%, 90% of breast cancer is. And when I did it, every cancer came back with a vengeance. 95 to 98% cancer incidence in my rats. Okay? Whereas if I gave it to cycling rats, and that's easy to do, all I have to do is surgically pop the ovaries out to make them postmenopausal, right? Very easy surgery. I give them to rats with intact ovaries. Tamoxifen killed all the breast cancer. Every single one gone. But if I gave it to an animal that doesn't have ovaries, everything came back. 3 years worth of work. And imagine, tamoxifen would have been banned all over the world. And that's $5 a pill. You think the pharmaceutical company is going to sit there? They would have killed me easily, right, if they could. Berkeley was protecting me the whole time, right? They wanted me to get fired. They wanted me to be, to quit. They offered me a job. $200,000 salary, and they said, we want you to be our consultant. You can continue your PhD at one of our approved universities. I knew exactly what they were doing. They were trying to get me off this research. I'm not getting off this research. It would have completely changed how we think about hormones. I could have been a father of a field. Okay? That's why research is 99% luck. Okay? And 1% hard work. So this is why PhD takes forever. Because it's all luck. Okay? So anyway, we can chat about my sorrow later. But I still ended up, I said, no, I'm not doing this work. I'm going to do something else. And I jumped onto vegetables and, and I set another record. I got 23 publications off my grad school. Okay? Nobody's done that. Still, the highest is 8. Okay? At Berkeley. So that was the highest record of publications. And 2 books. Okay, so everything worked there, and no company came after me because it was broccoli. Nobody cared about broccoli. But I still worked on estrogen receptors, and I still continue to do it. But every time I see tamoxifen, it makes me very sad, because that's what people get prescribed, and it's poison. It doesn't work. Anyway, so tamoxifen is an antagonist in a cycling woman, but it's a full estrogen agonist in a postmenopausal woman. Does that make sense? So it acts like a full estrogen in a postmenopausal woman, but in a premenopausal woman, it is an anti-estrogen. So it does work. in women who are still cycling and get breast cancer. So it works in them, but not women who get breast cancer after they're done cycling. And in the uterus, tamoxifen is a full estrogen agonist. It behaves like a full, so 95% of women who took tamoxifen came down with uterine cancer. Their breast cancer was cured, right? In 5 years, they came down with uterine cancer. So nothing like a bowl of broccoli. All it does is gives you gas. That's all side effects. You don't have any of this. The side effect of this drug is uterine cancer. Women are screwed both ways. Correct? You take tamoxifen for breast cancer, you come down with uterine cancer. Right? And then the breast cancer comes back.
Yeah.
So, I mean, how could they not think, right? And research was very improperly done in the 80s. That's the problem, because women's groups were pushing, they were lobbying for legislation to push this through the FDA as fast as possible. That's what happens when you do hasty research. This is what happens. You start seeing things you want to see. And then all the side effects start coming out, like termites out of the woodwork. Okay? So very important, the same hormone can have very different behaviors. And you know this, right? Neurotransmitters. Same neurotransmitter can cause smooth muscle contraction in one target and cause smooth muscle to relax in another target. Right? What's good for the goose, right? So why shouldn't hormones behave the same? In fact, they do. Okay? Synergism is when two hormones work together. And a paper from our lab, not my work, but a paper from our lab, showed that if you use tamoxifen along with our broccoli compound, it makes tamoxifen twice as effective in curbing breast cancer cycles. Okay? So women taking tamoxifen, right, it's bad, but at least you can take half the tamoxifen and supplement with half broccoli. Right? At least you're not getting exposed to toxic tamoxifen, right, at the full dose. So that's called synergism, when two hormones work together to produce a much greater effect. So hormone 1 has a little bit of an effect, hormone 2 has a little bit of an effect, but hormone 1 and 2, huge effect. Am I making sense? So that's called synergism. And they produce an added effect. Okay? So here's an example, right? So if you take cortisol, which is a glucocorticoid, okay, and then you have glucagon, which also raises blood sugar. They both raise blood sugar, okay. So if I took cortisol at a low, so this means I take nothing, right? This is my negative control, nothing. Then I add glucagon. Glucagon will raise it somewhat, correct? That's what it's supposed to do, raise blood sugar. If I add cortisol, it raises it somewhat, right? But when the two of them are together, whopping effect. Are we clear?
Yes.
So these two hormones are said to be synergistic. And they work by completely different mechanisms. Cortisol is a steroid. Glucagon is an amplified mechanism. Completely different. Glucagon is a protein. Am I making sense? Water-soluble hormone. Cortisol is a fat-soluble hormone. Right? So they work by totally different mechanisms. Okay? But they have the same added effect. Right? So that's synergism. Permissivity. Okay? This is the last one. So permissivity, what this means is I give permission for you to do the job. Yes?
Yes.
So let's say for argument's sake, right? So if you're a hormone, you're going to need your receptors to work, correct? Your receptors have to be there for you to produce an effect. Do you agree?
Yeah.
Yes.
Any hormone, doesn't matter what hormone, you have to have a receptor. Now, if I control your receptors, then I control your action, correct?
Yes.
So if I'm needed, okay, for, CAM's receptors to work, right, then I'm permitting CAM's action. Are we clear?
Yes.
So that's called permissivity. So when one hormone affects the level of another hormone's receptors. And the most classic is estrogen progesterone system. Estrogen will increase progesterone receptor levels. Okay? And so that's estrogen permitting progesterone to have a function, correct?
Yes.
So, in my opinion, that's all estrogen does in the body. Is progesterone is the main culprit in breast cancer. Okay, it's not the estrogen. And so what I did was I used tamoxifen because tamoxifen also increased progesterone receptors, right? And then I added progesterone. Breast cancer was back with a vengeance. Okay, and again, the person who discovered tamoxifen, he had zero answer to those results. Right? He just sat there going, "Oh, really?" I'm like, "Yeah, really." Okay, and these were human tissues, so I'm like, can't argue, right? Can't say that's a rat-specific thing. Okay, so estrogen increases FSH receptor in the follicle. So those are two examples of permissivity. Here's another one that I want you to add. Thyroid hormone increases epinephrine receptors. Remember adrenergic receptors? It increases epinephrine receptors in the heart, specifically. So thyroid hormone, people who have thyroid problems, that's why they're cold all the time, because the heart's not beating very well, because they don't have enough thyroid hormone. Because they don't have enough thyroid hormone, they don't have enough epinephrine action. Are we clear?
Yeah.
Yes.
What does epinephrine do? Raises heart rate. Correct? So if thyroid hormone is down, epinephrine receptors are down, the heart's going to beat very slowly. When the heart beats slowly, will blood reach the ends of your fingers? You're going to be cold all the time. Okay? That's why hypothyroidism, which is low thyroid levels, causes the feeling of cold, right? These people wear a jacket in the summer, okay, because they're so cold all the time. Okay? So that's another example of permissivity, right? Thyroid hormone is permissive for epinephrine to function in the heart. Okay? That's another example. All right, so we stop here. And the second part of this we'll do in lab on Thursday. The second part, today's lab already did, so you guys already covered this already. So the second part we will do in lab. Before you come Thursday, I need you to watch my videos. It really helps. Okay, otherwise it's a ton of material on Thursday, okay? So please watch my video and then come in so that the stuff will make a lot more sense, okay? All right, folks, so see you on Thursday. And again, reminder, next week's exam only covers
Muscle.
muscle, no endocrine. Okay, I'll go rewrite it.
Yes, sir.